Alright, so here's yet another video. What we're going to do is talk a little bit more about the role of microorganisms in the wastewater treatment. This is prep for day 6 on water filtration. Um, and so what we're going to do is kind of talk a little bit about that secondary stage of water filtration, the biological removal of contaminants out of water, and the role of microorganisms. And we're going to do a little bit of a review of some content to make sure that you understand the full context of what you learned last year and how we're going to connect that to this project currently. So, what I have drawn up here is an overview of some kind of reaction. It's the general process by which this secondary biological process of water filtration goes through. So we have this carbonaceous organic matter. So it's some carbon-based material that will then get converted with microorganisms. I'm just going to write that as MO, microorganisms, into gases and cell tissue, right? So these microorganisms are going to be processing some of that matter into gas, probably carbon dioxide as a release, and increasing their biomass, making more cells, more cell tissue, and increasing the volume and mass that those microorganisms take up. Now, there are a couple key words that I want to point out. So carbonaceous is similar to organic, and it's carbon containing. And for our purposes, this is going to be good enough for now. But there's another thing here, colloidal and dissolved forms of those carbon containing molecules. So colloidal or colloid is when there's a mix, like it's a non-crystal homogeneous mixture of one thing in another substance, and it cannot be easily removed. And when I say easily, it can't be removed with like a filter or with a centrifuge, like to spin it around really fast. These things won't come out. And so what the real goal here is to get this matter that's in the wastewater into microorganisms, get it pulled out of the water and absorbed into the microorganisms. And that's really important to think about. And oftentimes those microorganisms are bacterial. You spent some time looking under the microscopes at animals and protists like rotifers and amoeba, maybe some water bears. So there's some things that you're familiar with, but let's do a little bit of a review of the bacteria or the prokaryotes, prokaryotic cells. So what I'm going to do is talk a little bit about how we classify bacteria. So how we classify bacteria. So the classification of bacteria. So last year we talked a little bit about eubacteria and archaeobacteria, but let's talk about ways that we can classify them based on their needs and the way that they process matter and energy. So we can talk about one method here, or one system. We can talk about energy and carbon sources. We could talk about how they interact or their relationship to oxygen. And I should clarify now, based on our discussions of atoms and molecules, we're talking about oxygen gas, O2, or we could be talking about their temperature ranges. Okay? And so when we talk about those things, we should be thinking and reminded of a couple of key words. So I'm going to put a couple of keywords underneath that might jog your memory, and maybe some new ones here too. So we've got autotrophs. Remember, autotrophs make their own food. We've got heterotrophs. that eat other things. Their carbon sources and their energy come from other organisms. We've got these things called obligate. We'll talk about what that means. Obligate, O-B-L-I-G-A-T-E, aerobes. We've got obligate anaerobes. Should be thinking about aerobic respiration. Oops. Anaerobes. And we've got facultative types. We'll spell out what that means. For temperature ranges, there's some new ones that maybe you haven't heard of yet. Psychrophiles means they like something. Mesophiles and thermophiles. Okay, so what we're going to do is kind of define each of those so that you understand what each of those mean. But really the whole goal again is to convert some of that energy and the carbon that's coming in into macromolecules that the microorganisms can use. Carbon's essential for those building blocks, so we're going to make sure we understand the role of carbon in those carbs, lipids, and proteins. And that energy is coming in from some other source, whether it be light or some other chemical or some other organism that will then power the cell. So we have to remove both the carbon and the energy that's in the wastewater, right? So that's goal one, right? So this is the first goal, is to remove carbon and energy from the wastewater. That means it's moving into microorganisms. Oops, ran out of space on my board here. Microorganisms. But then the secondary thing to take care of in the same process is to remove the microorganisms. But you saw in the tertiary, or the third phase of the water treatment from Melissa, who came in for our day of lab. Okay? So let's talk about autotrophs and heterotrophs and different ways that organisms obtain energy, right? So in this section right here, maybe we'll call this section, I'm going to put a little dotted line circle around this. We'll call this A. So what I'm going to do is talk a little bit about A, right? So this is A. So let's talk about carbon requirements. And we'll talk about energy sources. Alright, so for carbon requirements, we know a couple of words here. We know autotrophs. Right? Autotrophs make their own energy, but let's think about what they use for carbon. Where do they get their carbon from? So think of autotrophs that make their own food. You should be thinking about maybe plants, some algae. They do photosynthesis or chemosynthesis sometimes. But in photosynthesis or even chemosynthesis, what is the source of carbon? Right? Where does the carbon come from? And you should know, you should hopefully remember that they use carbon dioxide or CO2. And that's their carbon source. Whereas heterotrophs use organic carbon, right? You use organic carbon. So that's the carbs, lipids, proteins, and nucleic acids. But for energy sources, we have another term. Instead of autotrophs, we have to be a little bit more specific here. We talk about phototrophs. We got autophototrophs. So phototrophs, photo means that they use light. And chemotrophs use chemicals, specifically glucose. They could use inorganic molecules, maybe even sulfur in deep sea thermal vents. Sulfur is a source of energy. So all animals, if we think about ourselves, all animals, we're an animal, are chemoheterotrophs. We can combine these terms, right? Chemoheterotrophs. Plants belong to the photoautotrophs. And so we could use that language and combine some ideas with one or two single words. Let's talk about B, section B here with the oxygen gas, relationships to oxygen. And what I'm going to do is circle this section, my dotted line, and call that B. We'll talk about B over here. Alright, so B, we have a couple of new terms maybe. So let's talk about what these terms mean. Obligate or obligatory. Obligate aerobes. So they obligate, or when you're obligated to do something, that you have to do something. So they must use or have to use oxygen gas, right? There's no way around it. So obligate, and the aerobe means oxygen, right? So obligate anaerobes. Obligate anaerobes do not use oxygen, or cannot use oxygen. Okay? And facultative anaerobes, so let's use this word, facultative. Facultative let's do anaerobes first. Anaerobes can adapt. Let's say that they can modify instead of adapt. We're going to use adapt for our own terms later. Can adapt or modify to anaerobic conditions. So they can change and be just fine. They can modify as an individual. And so that's very interesting to think about, right? So these things can actually change from anaerobic to aerobic. But when there's oxygen present, there are a couple things that die. Obligate anaerobes die. Sometimes they're poisoned by it, and obligate aerobes require oxygen to survive. I think it's kind of interesting. Alright, so the last thing I'm going to talk about in this video, I know I'm at 13 minutes. I'm going to try to stop here after I do this one. So this is C, and so in section C here, about temperature, we've got three new terms, and so what we're going to do is define each of those. So these are different categories for temperature ranges for the bacteria. So psychrophiles live when it's less than 20 degrees Celsius. Mesophiles, and there's a little give and take on this, right? That's where they're usually found, so less than 20 degrees Celsius. Mesophiles are typically in the 25 to 40 degrees Celsius range, and notice that there's a little bit of a gap in there. And thermophiles are in the 45 to 60 degrees Celsius range. And so there are all these different components that are required for this filtration system. You know that they were adding oxygen. We're bubbling that wastewater with the bacteria, so you should be able to identify what relationship to oxygen gas was there for those microorganisms. You might be able to determine which group the temperature ranges those bacteria fit into. You might have to do some unit conversions, but you should be able to figure that out just fine. Okay, so I'm looking at the time. I'm going to stop here. That's the end of part 1, Part 2 will be posted soon.